Hi, thank you for coming to the third seminar of the Precision Health Seminar Series. My name is Veronica Berrocal. I'm an associate professor in the Department of Biostatistics in the School of Public Health, and I'm a co-leader of the Education and Training Work Group of the Precision Health Initiative. For those of you who are not aware, the Precision Health Initiative is an initiative that was started about a year ago at the university with multiple goals, and the main goal is um, to facilitate interdisciplinary research across the university among researchers that are interested in precision health, and also facilitate events, education events like this one. We will also have a global precision health conference in May. They're also building tools for facilitate the interaction between researchers across the university that work in precision health. And finally, it's one of the goals of Precision Health Initiative is support the implementation of findings into real healthcare settings. Today, we have a seminar by Vera Paladanda Utapani. Vera joined the Department of Biostatistics this fall. We're very happy that he joined our department. He has been on the forefront of Bayesian research for many years. Before joining our department, he was for 13 years in the Department of Biostatistics at MD Anderson, and he was also a professor and institute faculty scholar at MD Anderson, and he held joint appointment at Rice University, Texas A&M, and the University of Texas School of Public Health. As I mentioned, Vera is very well known worldwide for his research in Bayesian statistics, Bayesian inference, and high dimensional data modeling. And he works with longitudinal data, cancer data. In terms of methods, he's worked with Bayesian graphical models, Bayesian semi and non-parametric models, and Bayesian machine learning. So, Vera, here is the stage.
Thank you, Veronica. That was a super generous introduction. No. Well, thank you for the invitation, and thank you for having me here and hearing my spiel here. So as I was pulling the slides together for this talk, so I reworked the title a little bit and thought I'll blend in a bit of a philosophical underpinnings and that with some of the technical, technical or more sciencey work, give a more high level overview of what we are doing, and give basically the bottom line is of this talk is a biostatistician, or more broadly, a quantitative analyst or a quantitative scientist point of view to big data integration and those sort of things. Okay, and a biostatistician angle, if I had to put it, in an accurate manner, is with the deluge of data with that's coming from all angles, collaborations, and that's a happy me, actually, by the way, I should say. I'm smiling behind that because the more data, that keeps us in business, and that's why we have a job. So, no. So to set some context, I think there's various variations of this slide that you've seen in many talks, or what's the levels of data that's coming from different angles, and there is a lot which is in terms of structured data from some molecular space, the genomic space, but also from unstructured spaces that includes imaging, biosensors, and social media or internet, those sort of things that are increasingly unstructured data that's getting digitalized and the use. And I think the key goal is sort of a very high level key goal is sort of to extract some biology in the context, some context to it, and also the eventual goal is having some clinical implementation or clinical relevance, I should say. That's probably a better word here. Okay, so I don't think this is an understatement as yet, but it is, from my perspective, it seems data integration may be the key quantitative challenge for modern science, given where we sit in terms of the data generation. Okay, and I thought a little bit about this, and there's a bit of an art and science about it. In the art comes from the ingenuity and the creativity, how to put it together, and the art is, So if I had to put up this picture, this is a very famous picture. If anyone tell me what this is, no? Anyone? Exactly. That's, it's a famous. It follows from the famous quote of Aristotle. It says the whole is greater than the sum of parts, and so what's, so the key idea is that each different data offers a different insight into the underlying biology and gives an incomplete picture. And the scientific aspect of this is how do we link it all together, okay? For different types of information, different types of data to get a more holistic picture across human diseases in a biomedical context, all right? And to tell you the importance of the problem, the pertinence and the importance of the relevance, it's a pertinent problem. These are two issues, and these are not relatively old, So, in terms of where the field is moving, in terms of, and there's much more, over the past here. So I want to talk about the cancer context because I used to work at MD Anderson Cancer Center. I spent most of my career there, 13 years there now. So it's a disease I know a bit about, And so there are many bad things about cancer. But one of the good things from my angle is it's a very well studied disease system, okay? And that makes it a very germane area to do sort of the integration. So the integration type I'll talk about, so to set some context. So also, not only from molecular angle, from the biological, from the genomics angle, but also clinical and environment. So a lot of resources have gone towards it in terms of funding, and it's well studied, okay? So, the types of data that's coming out includes not only the high-dimensional genomics data, and this was a big consortium level efforts. I'll talk about it in a minute. But also, because the technological advances with the genome, the individual labs can generate massive amounts of data, on multiple samples, and that's more institution specific, but all collated. It's growing day by day as we speak, right? So when I was at MD Anderson, I had a good fortune of working closely with the TCGA, which stands for the Cancer Genome Atlas. And this was a project that was launched, I want to say mid-2000s, and has collated over 10,000 samples with detailed molecular data at different levels. And I saw sort of in the trenches how this data, because MD Anderson being a high volume patient cancer center, contributed a lot of samples and sort of questions to just understand. So there are papers that are already published, like path-breaking papers, more being written as we speak, all right? So there is, when they collected all this data on 10,000 samples, I think to set up some context, there's sort of two competing continuums here. One is sort of because it can be easily measured in some sense, and on the other continuum is establishing some sort of relevance. So this is sort of reiterating the same point in the context of cancer, is how do we, a single type of data tells one part of the story, and how do we basically gain two levels of understanding. One is just systems level understanding, just what's going on, what's biologically, what mechanism changes. And then at a translational level, keeping that context in mind and correlating this with clinical outcomes. And I guess that's what links it up with the precision personalized medicine paradigm, which on a very high level is basically taking all the data together to, so there are various nomenclatures of it. Precision personalized medicine, it's the precision medicine, there's some people like personalized, some stratified medicine. And the key idea being using all this data to basically break apart subpopulations with the, oops. That was supposed to be, sorry, a survival curve where, raising basically the tail probabilities of how long a patient will live, okay? And figuring that out, okay? So from a quantitative analysis channel, there are various challenges to it as a starting point, okay? So a first degree challenge is, of course, the storage and retrieval, storing this data. And it's a more, say, a step zero process, which is more from an informatics perspective, okay? And another main thing that before any downstream analysis, it's more how to get the data quantified, and does that data reflect the actual biological process? So that's a pre-processing normalization, just the quantification of the data. And I feel this is absolutely critical, and most statisticians have sort of sat on the sidelines for this in terms of thinking about the design aspects of it, the data imperfections, and just the matching of the samples, and what sort of artifacts could overwhelm the actual biology. Okay, and also thinking through what might be the scale of the data that we are looking at. Scale in terms of not the magnitude of the data, but just how it is measured and what does the quantifications actually mean, and setting that. Okay, and from a statistician's angle, from biostatistician, from computational, it's an ill-posed problem in some sense, because even though the sample size is increasing, we are still, at least in the context of cancer, it's still relatively low, hundreds to thousands, as opposed to GWAS settings where tens of thousands of samples can be sampled. It's an ultra high dimensional situation, and not only with the number of variables, but when you start considering interactions and such, second order, third order, and these where the classical approaches fail. And I want to add one more to this list, and it's in terms of what I call structure dependencies in these data, so which is not straightforward. So what I mean by structure dependencies is dependencies that sneak into the data, and that can be induced multiple ways. So it can be broadly classified in two ways, and the broad buckets they can be either just the biological dependencies, just the way the data manifests itself. So it could be serial genomic correlation when it's like copy number or SNP arrays, or it could be pathway based when you look at higher level gene expression or protein expression, or there could be mechanistic dependencies, as DNA goes to RNA goes to protein, and nonlinear interactions. And the second, which is often critical, is also how actually the data was collected. What was the experimental design? What was the sampling, the random sampling? How was the sampling done? So increasingly, at least genomics data comes from a sort of a secondary analysis of bank tumor samples. So these come from, say, there are two major landmark trials called the BATTLE trial or the I-SPY trial, which were adaptive trial of different drugs, regimens in lung cancer and breast cancer. And as a correlative study, they take large amounts of gene expression or other platforms that are measured, and these biomarker based. So there's an inherent, a subsetting of the data that happens at there, and that needs to be accounted for. And increasingly, there's a lot of data that's coming in terms of spatial characterization of tumor development, saying that one biopsy might not be enough. We need to get a whole picture of the tumor, so we should sample different areas of the tumor to get and also imaging that sort of. Okay, and the key goals are how can we incorporate these sort of dependencies in principle statistical, of course, but more broadly analytical frameworks. Okay, so as a statistician and as a Bayesian statistician, I guess I'm going to make a case for model-based analysis and why we need to think models when analyzing this data. And this is a more philosophical point because it's a biological system. We have observed empirical data, and we want to theorize them with certain amount of models. And there's an assumption there, but there are a lot of advantages of going the modeling paradigm as opposed to, say, algorithmic or summarization paradigms, is basically the incorporation of structural assumptions. The structural assumptions in the data can be incorporated for. And the borrowing strength of different sources can be done efficiently for better signal detection. And then it also allows scientific, because models have parameters. I'm talking statistical models, they have parameters, and parameters mean something. So there's explicit scientific interpretations that are linked with it. And then this is probably the couple of key points, is the quantification of uncertainties. Now getting estimates and what's the uncertainty, and accounting for the different sources of variation, and also majorly being the multiplicity controls, and how can we do that in a coherent, and that's really critical for high-dimensional setting, especially for validation studies. okay? And, especially Bayesian models can incorporate prior beliefs about the model, and those are especially amenable, okay? I think one drawback could be the scalability or the computational tractability of these. It's a work in progress, but still we've gotten very far, and it's sort of doable with the current computing infrastructures you have here. Okay, so to give you just examples, so when you start thinking about processes, so the models that I'm talking about, process models. So as soon as there is a quantification of the biological hypothesis or a fundamental hypothesis, you can link it to a specific class of models. So you start thinking predictive prognostic, you start thinking sparse regression models, you start thinking discovery of subtypes, it's naturally clustering models. And when it's pathway-based, it's more graphical models or network models. Then you have mechanistic, then you start thinking there's a hierarchy in there, so DNA to RNA, RNA to protein. And then when you have nonlinearity in the system, you expect or you hypothesize a nonlinearity in the system, a non-parametric and different functional data spatial models. And then we're going to combine everything now, integrated models, okay? So that sort of was the 70,000 or even 100,000 foot overview, setting some context. And I'll tell you a little bit, give a flavor of what sort of things I've been involved in with different groups, and is talking about basically three different streams. And I'll talk about hierarchical models, how we think about integration. Specifically, I'll talk about something we call the IBAG model that we developed for integrating multi-platform genomics data. And then I'll talk a bit more of the recent work that we've been doing in terms of looking at network regression, and especially looking at what we call personalized networks in cancer. And that's something, a pet topic of mine right now that I'm really, really excited about. And then how do we combine genomics with imaging data? And pretty much everything is in the context of cancer, but the methods as such are broadly applicable to a variety of disease areas. That's right. Okay. So when we started thinking about data integration problem, this was, I guess, 7, 8, 10 years ago. So we started to say, what's a good starting point? And we set the data as this sort of a flow diagram, where we conceptually thought that we put all the platforms on one side, and then all the outcomes on different. So this is sort of a first-degree skeleton starting point. Okay, it's a crude starting point, but it's at least somewhat justified. So you have the genomics, and that affects the expression. And the reason we do that is there's this theory, at least in cancer, that a lot happens at the DNA level. That there are different kinds of DNA level due to genetic inheritability that you've inherited. There are environmental factors, but when you come down to expression, there is sort of a programming, or the oncogenic programming, that is sort of stable across cancers, across patients. And the chief examples of that are, for example, in breast cancer subtypes, the HER2/neu, and then those have been discovered by expression data. For the glioblastoma, the subtypes of glioblastoma have been discovered through expression data. So to leverage that, we take the expression as the central piece, and then, okay. So way we build is the upstream platforms, the different molecular data. There's some sort of a mechanism between them, and then look at the clinical model, and which links those to the clinical outcomes, the relevant clinical outcomes. Which could be, you can think about survival, or it could be also imaging, or any outcome that's patient level that you see. Okay, and we just basically put the 2 and 2 together. Okay. So as I said before, so we take the gene expression as the fundamental unit. So saying this, we observed a gene expression, and then we use the upstream platforms, which is say, in this case, think about just copy number and methylation, and we break that into components that's regulated by these upstream platform. Okay, and we do a decomposition, and you can think about gene expression as a summation of all that happened before it in terms of copy number and methylation, and there are 3 components. So suppose this is 100, you break it down into, I don't know, 40, 30, 30, for example, okay? So, and then you can think about this as the denoising, and then you combine them with the clinical outcome and to link it to the clinical outcomes, okay? And this work, most of it is prepared by one of my graduate students, past graduate students, Elizabeth Jennings. Okay, so this will probably be one of my most mathematical slides. So I'm a statistician, and I have to have some formulas in there to get into my comfort zone. So basically, the way we do this is, I'll probably give a high-level summary here. It's basically we take the gene expression and use the upstream platforms to run a nonlinear regression model. And so we don't expect the relationship between the expression and upstream platforms to be linear. So we let it be nonlinear. Let the data tell us what the shape and effects are, and then we collate these effects right here, and those becomes the denoised gene expression contributed through various upstream platforms, and then we plug it into what we call a clinical model. Okay, so we have gene expression, we broke it down, and then we call it. Okay, and why do we do that? So this sort of came about at various discussions with a lot of my collaborators who are physicians, scientists, and the key idea here is what would be a good gene as a downstream. So this is from a more discovery angle. What would be a good gene that's either which can be validated either preclinically or clinically? What would be a good candidate? Okay, so if you think about genes, the cells and cells within, cells have DNA, and then something goes wrong in that gene. Okay, so if you think about it like a switchboard, and it had various switches, like it had an epigenomic switch, a genomic switch. So what switches possibly went on or off? Okay, that conferred something that gene, something went wrong with that gene that conferred an advantage or a disadvantage to an outcome. So if you think about survival, a patient lived longer or shorter. Now, what contributed to that? What contributed to those effects? Okay, so if a gene, if we can find mechanisms of those switches or mechanisms through those switches, that will tell us a deeper into. So if a gene that's really screwed up at multiple levels, at the DNA expression and methylation level, becomes a higher candidate for a future validation study. So basically, this method discovers those genes and what was the mechanism that had an influence on the outcomes. Okay, and it doesn't have to be mutually exclusive. So a gene could be, could have all three switches turned on or a combination of any of the two. Okay, so so just as a case example, we worked with the glioblastoma data, which at that point was one of the first cancers to be assayed by TCGA, and had grown to a very high. I think it's closer to 1,000 at this point, and once you start matching, you have around 280 patients. Okay, so what sort of outputs or benefits are there of doing this modeling? Is, as I said before, it spits out. So here is a you can think about this as the probability of selection. So the higher the bars, the more important is the gene. And each gene, if you see, it has 3 bars here. So basically it tells us, so these are genes that have a positive effect on survival. So these are the good genes in some sense, if they cross that threshold. And then, and each of the color represents, was that gene affecting through methylation, copy number, or other, I forgot to mention, sorry. The other here is aspects that we didn't measure, because the gene expression can be influenced by multiple factors. It's just not copy number and methylation. There's a lot of transcription factors or any other factor that we don't know about that could be, or purely transcriptional that we don't know. So we let the other be in the model, so it's not pure noise, to find out what other mechanisms, unexplained mechanisms, at least from the data we have. And the other benefit is, like, if indeed there is a strong dependency between upstream platforms and gene expression, you get much better signal detection by borrowing strength between the 2 data sources rather than not. And that we showed both in in this in the specific examples, but also through simulations and such, just on synthetic data to establish that. Okay. So, so this has been a series of works where so IBAG, it's not just for gene expression. So it's the construction is pretty general that can integrate arbitrary number of platforms. You could have methylation to the mix and have microRNAs and then mRNA, or you could do DNA, mRNA, or protein. Now, or we have recently extended it to PyBag, which we call, here we think the genes are independent, but we add in the functional pathway information to look at gene sets. And then a further development is, we just got this paper accepted, is how to include radiographic imaging and multi-platform genomics into finding these mechanisms. Okay, so I want to talk a little bit about PyBag and how do we extend that. And so the main motivation here is it's taking into account the higher order organization of these genes. So it's well established that gene sets are nested within pathways, or pathways are nested within platforms. So if you think about gene sets, now these are pathways. So typically, most drugs are targeted towards pathways rather than single genes. So finding these gene sets. So PyBag is a generalization that not only integrates multi-platform genomics, but also it flags genes within platforms, then ranks the pathways that are important, and also finds out which are it also identifies the regulating platforms for important genes and pathways. So it does this hierarchical procedure where it gets to the prognostic, depending on the clinical outcome, the prognostic ones. All right. So this is an example plot from PyBag, where here we have plotted the pathway, what we call the pathway scores. So the higher it is, the more important it is. So these are pathways from GBM data. So if anybody's worked with GBM or even cancer in general, you see some pathways are pretty fundamental here in some sense, especially like the PPAR pathway or the JAK-STAT or the P53 here, and they rose to the top, and also looking at different mechanisms that underlie. So these are the top prognostic pathways that affect the genes. All right. Okay. So there's an app for everything nowadays. So when you start this, we start putting, this is the, we have an IBAG Shiny app, and it used to be deployed on my MD Anderson server, so we are in the process of moving it to the Michigan server, hopefully soon. And okay, so I want to switch gears a little bit here and talk about network-based analysis, and give the work we are involved in, and what's the next steps. So at a very high level, I guess networks, the basic premise here is getting a more systems level understanding. So I was talking in PyBag, these gene sets there's a higher order organization to these genes, so understanding that, and also instead of looking at one or two genes or proteins at a time, how can we go at a more global level to get. So in the context of integration, data integration, or genomic data integration, I think there are many inferential problems, and some of the inferential problems that we are tackling is looking at multiple types of systems, which not only includes the patients, but also cancer cell lines, the idealized cancer cell lines, and more recently, the patient-derived tumor xenografts, which are called the PDXs. So PDXs are models where you take tumor from an actual patient, put it in mice, and passage it multiple times. So cell lines are grown on a petri dish, so they're sort of idealized systems. But PDXs, the tumor is inside another organism, so it's a living, breathing tumor. So it's sort of more closely related to the pathophysiology of an actual tumor from a patient. And then you can do drug testing and everything on PDXs. So we are leveraging all three, and then also looking at the entire omes to sort of not only find what is the mechanism of dependencies through different kinds of graphs, but then in terms of finding which graphs are different or which graphs are heterogeneous, and also patient-specific networks, and I'll come to that, and build prognostic models based on these networks. Okay. So a couple of works in this is in the realm of what is classified as vertical data integration. So what that means is basically it's the same samples. So the samples are the same here. The genes or units are different here, but you have three different platforms. And finding relationship between the three, and they're matched by the samples. So that's led by one of my former graduate student, Yang Ni, now at UT Austin. At Texas A&M as a junior faculty. And the other is horizontal, what is called the horizontal data integration. So it's basically looking at the multi-platform genomics, but now actually going across cancers, so what I call the pan-cancer models. Okay. So the key idea being if there are some cancers which have some similarity, so the similarity could be through the site of origin, for example, GI cancer, so they could be in the abdomen area, or it could be, say, female cancers or women cancers like ovarian, breast, and uterine cancers. So can we understand the biology that's common to the three cancers in terms of not only what is conserved across the cancers, and also what's different among them, and how are they linked together, okay? Which can tell us something about the shared biology. So it's basically thinking towards, can we do discoveries, this increasing in what is called the basket trials, where you actually don't treat the organ, you actually treat the mutation or the gene. And the gene could be significantly disrupted in any of the cancers. So rather than treating it, so it's going beyond the organ to the gene level. And that's the hope with these pan-cancer models, okay? And so these are some of the works that over the past few years that address these different aspects. And mostly we worked with TCGA data because it's such a well-curated database in terms of not only getting the data, but also it's analysis ready in some sense. And there's a lot of, it saves a lot of time as an analyst in terms of getting those, okay? So I'm going to spend the rest of the time on talking about what is called the personalized networks in cancer. And as I said before, this is a topic I'm really excited about. It's and the basic idea for this comes from what is called tumor heterogeneity. Okay, so tumor heterogeneity in essence means that that it's assumed that cancer evolves. There's a population level commonality towards that, but it's increasingly becoming the things we're increasingly finding out that there are also groups of patients that are completely different. There's an inherent differences, and can we leverage that information? So in this case, you had a population of patients, and if you break it up into multiple populations, each having its own sort of a disrupted network. And going beyond that, instead of finding groups, we thought this should be a little bit more general, because there could not be a strict barrier or strict way to define these groups and have that in a more fluid manner. So if you could think about ranking this patient as some sort of disease evolution, can we build networks or some sort of metrics of disruption of these networks or pathways as the disease evolves, okay? So there's no one breaking point, but there could be multiple breaking points along the disease evolution towards cancer, okay? And this disease evolution can be defined in multiple ways. It could be the standard staging of tumors, like the blood biomarkers, or it could be the stage of tumor, or any way you can define them. So can we let that disease evolution? Okay. So to this end, we have developed a method. It's a Bayesian method called personalized cancer-specific integrated network estimation. We call it PRECISE. It took a while to get the fun there, but also we actually reworked the title to get PRECISE. But yeah. So the basic idea, and this work was spearheaded by Minjin Ha, who's a former postdoc and now an assistant professor at MD Anderson, and basically did almost all the work here. So the basic idea here is so we looked at proteomics data, what is called the RPPA data, and combine it with different levels of upstream data with methylation, microRNA, and mRNA expression, and also look at prior information that are stored in these databases, because a lot is known a priori about these protein data. And the idea being that can we leverage that prior information and build networks that are not only cancer specific. So we looked at around, I think, 33 types of tumors. So build networks that are cancer specific, but from these cancer specific networks, deconvolve somehow subject specific networks. The networks that are subject specific, that could potentially, in this work, we let the network connectivity being shared, but the nodes are, the node attributes could be different, or the nodes can be, expressed in different ways. Okay, and then find metrics that we can somehow collapse this whole network into finding a sort of a one summary score that we can classify these networks. So if you think about these networks as for a given pathway, you think about pathways and find these pathway scores as suppressed, activated, or neutral, and doing this in a totally unsupervised manner, and then look at identification or outcome prediction. And okay, so basically it's a Bayesian model that goes through and does the estimation in a network sense. But let me get to the results. So let's see. So yeah, we did 32 tumor types, and the results are in the paper. So this is one plot from the paper, and that sort of gives the idea. So so you have the raw matrix of the entire protein and by samples. So this is around 7,000 samples, and this had around, I don't know, 300, 400 proteins. And what this is collapsed to is 12 core pathways, okay, that are most representative in that data and in cancer. And what's plotted here is an unsupervised heat map of the activity of those pathways. And if you see this top row here, that's the lineage. So that's the known lineage of the cancer. And the below is the cluster that we found. Okay, so it's not super unsurprising, but in some sense, because there's a high fidelity to the tumor site of origin, which is understood, because that's what the biology is capturing, but a highly processed summary of the proteomic pathway information can have so much information to have so much specificity to find so much fidelity to the tumor site of origin. And if you plot just the raw data, the RPPA data is not as clean. So there's a lot more mixing between them. Okay, and we looked at the prognostic power of these signatures too, and it had higher prognostic power as compared to other approaches that combine these pathway activities. And so in the 3 minutes, so 5 minutes I have, I'll talk about, so this was more in the discovery domain. So one thing we started pondering, or rather I started pondering about is how do we establish the clinical relevance of these discoveries? And as a sort of a quantitative scientist, basic scientist, how do we do that? So where are we moving with these things is going more generally. So looking at, so right now we're looking at just 2D genomics. So we have genomics and then we have conditions, but can we move beyond that and get in the drug information? So if you think about this as 3D genomics, so can we get doses, can we get longitudinal profiling, and can we integrate that levels of information into the and look to integrate those kind of models, and what might be, what sort of information can we gain? And so this is one of the current projects that we are working on. And so this is a busy slide. I can walk you through it in a nutshell. So basically, the key idea here is how do we match the right patient to the right drug? Okay, and so the ideal way is, of course, doing a randomized controlled trial. But can we leverage information stored in these cell lines, which have a very rich drug profile stored in them? And then if we know the genomic similarity, can we use the patient's genomic features to correlate with the cell line genomic features, and then build a model where we know these cell line drug responses to sort of calibrating what could be a potential therapeutic response for a given drug. Okay, so the key here is take the genomic features. So there might be some patient who will be mapped to some cell lines. Okay, so find the idealized system that can be reflected. Like if you think about genomic features being sort of the fingerprint of the disease, so can we match those fingerprints, the high dimensional fingerprints, so we could use all the available data to get the closest match. And finding that metric of match, that's inherently an analytical problem, finding that match. And then taking there and then filling in this missing link of finding the perpetrator. So, and I didn't put it here, but we ran this. So we ran this method on 2 data sets where we hid these therapeutic responses in myeloma and I think it was breast cancer. And then we looked at what might be the drug responses had we gone through and calibrated the therapeutic indices for these patients. And we saw that it provided better separation in the trial, and if we don't incorporate the cell lines, just training a joint model on the patients and cell lines together to get these drug responses, and that's what we call an IRX model, an individual therapeutic responses model. So it's still a work in progress. It still needs some rigorous validation, and our idea here, our future work here is basically not only you look at clinical trials where there's data available on the patients in terms of therapeutic responses, but go beyond that. But look at the TCGA cohort and see if can we do some repurposing of some drugs or know some therapeutic index defined by the genomic profiles. And that could lead us more insights of what kind of drug therapy could be potentially valuable from a discovery. Okay. So we have a Shiny app for the Precise. And so I'll probably spend a minute defining the problem in radiogenomics and telling a little bit about how to incorporate imaging data. And this is it's a good and a bad problem to have. The good problem being that the key idea here is to get molecular data from patients, there needs to be an actual biopsy. So you need to stick a needle in and get the materials out. But on the other hand, imaging, it's non-invasive. Okay. So from the same patient, we can get an idea about what molecular markers are there of imaging that can serve, and if they have a good synergy or good correlation in some sense, then that can be looked upon as something called a virtual biopsy. Okay, because the imaging marker is a surrogate of the molecular marker. Okay. So that's the basic problem, but the problem here, at a very statistical angle, it's how do we do population level analysis? And I'll get to the picture here that might explain it better because the tumors they grow in different parts of the brain. So this is GBM, This is GBM, and if you see here, the tumors are different sizes, they're different shapes, they're located in different sizes. So how do we get a common mapping of these tumors so that we can do population analysis? So in imaging analysis, typically it's like fMRI data or either data. The matrix is conformable. So if you conform to a common template, voxel ABC means the same across the individuals. But in this case, getting a conformable template is a very hard problem. So how do we get these metrics? And also these metrics that can capture this architecture of the tumor development. Now, so some of the work we're doing in our group is basically defining these different metrics. So it could be the shape, it could be something called the voxel density estimates, or it could be just looking at trees and tree structured data. And so this follows from basically the core paradigm of how cancer develops. So the cancer actually is known to develop that it's one cell gone wrong, and then one cell goes wrong and confers a local advantage to the cell right next to it, to the next to it, to the next to it, and then the cancer spreads. So if you think about the voxel level information being a cell, so this tree pretty much captures that everything was one cluster, and then it broke down into different clusters. And the busyness of the tree, rather than how many clusters it broke apart, kinds of tells you how heterogeneous the tumor is. So in this example, this is a GBM example where this patient was is a long survivor, around 60 months, and this one was a short survivor, and 1 month, and this is probably a superstar because the average median survival time in GVM is 12 months. So, and you can see that this sort of recapitulates the tumor heterogeneity. So the topology of the tree manifests the tumor heterogeneity, the different structures. So it turns out to be a cute analytical problem. And how do we do, analysis on tree structured data? Okay. So a few summary points I would like to make. It's a very exciting field. Integrative analysis. There's a lot of still computational and statistical challenges now because technology evolves on sort of a very rapid pace, and there's a lot of a plethora of analytical problems in this area, and also looking at the other type of genomes, and beyond cancer, looking at the microbiomes or single cell, or looking at CRISPR, and basic idea being how does genomic diversity affect the phenotypic diversity? And then, like I said before, we are looking at more pan-cancer models, so you can get much higher sample sizes and exploit these commonalities. And then, of course, I think one of the facts that needs to be grounded in is of course, going from discovery to translating. The holy grail still remains. How do we do, the validation of these things? And I think that's a critical piece, and but we are taking some steps towards that. At least prioritizing what can be done, because these are expensive experiments, and also incorporating population health data resources. How can that be leveraged? Right. So I want to acknowledge actually my collaborators, and especially my students and postdoc who pretty much did the work. I just give talks and get the money. No. So I want to spend a minute on the background picture. This is actually, most of this work was done in MD Anderson. I just got here 6 months ago. And if you see here, this is where the biostudy used to sit, and this is all my collaborators who used to sit. And you can see a bridge here. It's a literal and a metaphorical bridge that had to be crossed multiple times in a good way. All right. Thank you.
So thank you, Veera. We have time for questions. If anybody has
Yes.
In the pre-targeted treatment age of cancer treatment, we had a pretty small number of cytotoxics, chemotherapeutic agents, that some worked for a random group of 3 or 4 tumors, others worked for a random set of 5 or 6 tumors, and we never really figured out why at all. Now that you have gone through the TCGA and mapped the similarities and differences between 30 different tumor types, and we know the genetics within those tumor types, shouldn't you be able to overlay the hundreds to thousands of clinical trial results and try to figure out what are the patterns that cause cisplatin to work in a couple tumor types versus not in others, et cetera, and then possibly use that to select chemotherapeutics for patients based on their specific profile within a cancer type?
Right. That's a great question. That's a great question. And when I talk about that IRX model that we were trying to do, it sort of goes into that domain. I think the critical problem is having clinical trial data which has a rich enough genomic information for us to validate, first of all. Can we know that these were the responders, these were the non-responders, these were the genomic profiles of the. So the TCGA, I mean, it did a lot of good things, but one of the bad things is there's no good curation of the prior treatments. So we don't know what the patients were treated with, even within tumors. Some of them have, it's higher in some tumors and lower in others. If there was a good curation of the clinical data, that would help us a lot. But the problem is in TCGA, that didn't happen. So we had to go in for this part. For the IRX, we had to go into, we found only 4 trials that had trial where they had curated gene expression data as part of a randomized trial that was out there for us to repurpose those drugs. And that's exactly where we want to get, and to say if we can find out, if we can repurpose that drug and see it might have worked on this particular subpatient. And that's exactly what the analytically we are trying to do.
Even without the individualized tumor genomic information, just knowing across that tumor type, these are pathways that are more often switched on or off, or these are variants that are more often in, because once you're subsetting them, it seems like you should still be able to pick up some patterns and then go back to the national cooperative groups and say, we want to validate this in your clinical trial by actually analyzing your samples.
Right. Yes. That's no, it's a great point. But exactly that's where we would like to go, and do that. No.
Other questions for Peter?
I think it's a very promising approach. I have just a little technical question about the precise study. So you're a Bayesian statistician, so I expect that all your studies, you somehow show that Bayesian approach is superior to other approaches, for example. So I'm wondering whether you could comment on the kind of power of Bayesian modeling in these integrative studies. So does it help to generate more robust like a clustering, for example, in the heat map you showed?
So that I'll give you two answers to that question. So one is the why Bayesian helps, and more in generally modeling helps, because there's the method. The Bayesian one helps us because we're bringing in the prior information. We know that there are some protein-protein interactions, and how do we incorporate it? I think it's much easier to do in a Bayesian setting, and that prior information helps us. And the gains that we see in terms of the clustering are due to bringing in the prior information. But the second question is more high level. It's the modeling, that we can leverage. The basic question we answer there is what is a metric of pathway activity? And instead of, say, for a given pathway, either summing the protein expression or can we get a network of that pathway and use that information? So the way we did it is basically we took the nodes in the network and say what are the other nodes it's controlling and getting controlled by. And if there's a node that's controlling a lot of other nodes, that's an important member of the pathway. So if that's switched on, it should get a more higher weight than the other nodes. So, intuitively, that's how we build this pathway score and got the node activation, got the pathway activation statuses, and then we correlated. So it goes more like, instead of doing just, using summaries or just doing some modeling that can coherently represent some prior belief, that helps. And so we did only empirical studies in this data set. So it's better clustering in terms of better clustering. So how can you define better clustering is through how much fidelity is that to the tumor site of origin. So that's one. How is the prognostic? So if you have survival outcomes, does that do better than any other method? Non-network method or non-integrated method. So that's what better one. No. So it's just the validation of the method as no.
If not, let's thank Peter again for a great talk.
Thank you.
Okay, so our next event is February 13th. The speaker is Minoli Pereira, who's an associate professor in pharmacology at Northwestern, and she will speak about pharmacogenomics and genetic variation in drug metabolizing enzymes in African Americans. So thank you so much for coming, and look forward to see you next time. Thank you.